It's written no internet.
Dr. Saeed, should we start?
Yes,
Or
please.
wait?
Please start, please.
Assalamu alaikum wa rahmatullahi wa barakatuh, and good afternoon, everyone, and welcome to our third Research Cafe session. First of all, thank you for joining us this afternoon. It's our pleasure to have you all with us as we come together to explore and discuss current research and innovation in health sciences. Today, we are pleased to host presentation entitled Reduced Expression of Co-regulator TLE1 in Type 2 Diabetes is Associated with Increased ILS Alpha Cell Number. Before we begin, I would like to introduce our distinguished guest. First, we are honored to have Dr. Shadi Al-Bahlani. She's joining us today as a research and topic expert. Dr. Shadi Al-Bahlani is an associate professor in Department of Biomedical Science, College of Medicine and Health Sciences at Sultan Qaboos University. She obtained her PhD in cellular and molecular medicine from University of Ottawa. She is currently heading the Quality Assurance and Academic Accreditation Unit in the college. She has a long and wide experiences in the institutional and medical program accreditation, as well as curriculum design and review. She is as well an external reviewer for the Institutional Standard Assessment with Oman Authority for Quality Assurance of Education. Dr. Shadia has a research interest in cellular and molecular pathology, mainly in cancer. And it's our honor to have today Dr. Shadia with us to share her expertise and experiences in the topic of research. We are also delighted to welcome our presenter, Dr. Najwa Al-Jahdami. And Dr. Najwa actually is a well-known person among us. She is a PhD holder from the University of Newcastle. And her research topic mainly is on diabetic mellitus, as her thesis was titled as Improvement of Culture Condition in Pseudo and Human Islets Toward Improved Islet Transplantation Outcome in Type 1 Diabetes mellitus. She has long experience at OCHS, and currently she is working as program supervisor of MLS program. She is also a member of Research Central Committee at Ministry of Health, including diabetes at United Kingdom. She is also certified as an external reviewer of OAAA and Quality Assurance of Education. She has published several studies in diabetic mellitus internationally and locally. Before handing over to Dr. Najwa Al-Jahdami, I would like to briefly go over a few housekeeping rules. So please, and I would like all of you to cooperate with this so that we can get a fruitful presentation. Kindly please keep your microphone muted throughout the presentation so that we can minimize the background noise. And also, if you have any question, post them in the chat box as it will be addressed at the end at the discussion session. And I would like to request all of you and encourage you to stay until the end of the session and actively participate in the discussion. Thank you initially for your cooperation. And without further ado, it's my pleasure to invite Dr. Najwa to begin today's presentation. Dr. Najwa, the floor is yours.
Assalamu alaikum wa rahmatullahi wa barakatuh. Thank you very much, Dr. Fatma, for the introduction. And I would like to thank also Dr. Shadi Al-Behlani for being able to take part of this presentation after a struggle with trying to find an expert due to summer holidays. I would like to thank also everybody who attended the presentation. I would also like to thank Dr. Saeed Al-Rawahi for his commitment to run this research cafe smoothly. I will go ahead and share my screen and I'll start. So let me just share the presentation. Is the presentation clear to everybody?
Yes, it's very clear, Dr. Najwa.
Okay, thank you very much. I'm going to go ahead and start with my topic today, which is, one second, I think I'm just making the screen wider. Okay, so today I'm going to talk about the reduced expression of the co-regulator TLE1 in type 2 diabetes mellitus, which is associated with increased islet alpha cell number. So basically, this is a picture of an islet, as you can see here, and it is stained with different immunofluorescent stain because the topic is about islets, so I thought that would be interesting to see. So today what I'm going to go over is an overview of diabetes mellitus and islets of Langerhans, because most of the time people don't know what is islets of Langerhans in relation to diabetes mellitus. I'm going to look at some cellular dysfunctions in diabetes type 2 mellitus. Then we're going to go to explain the role, which is the major transcription factor here we're going to talk about, which is transducin-like enhancer, split TLE, which has a role in the fate of beta cells. Next, I'm going to explain the aim of the study, methods and results, and finally, some concluding remarks. So let's just have a recap of diabetes mellitus. I'm sure everybody knows what is diabetes, but just a simple recap. Diabetes is a group of metabolic disorders. We all know it is characterized by increased glucose level in the blood, which is hyperglycemia, the scientific term. This is resulting in defects either in insulin secretion, insulin action, or both. So we have majorly two types of diabetes. There are other subtypes of diabetes, but the two major types are diabetes type 1, which is characterized as juvenile diabetes. And we have diabetes type 2, which is adult diabetes. Majority of cases compromised in diabetes type 2, about 80% of cases due to lifestyle. So today my focus will be diabetes type 2 mellitus, not diabetes type 1. And when we look at the difference between the two, diabetes type 1, as you can see here to the left, is because autoimmune...
Dr. Najwa, sorry for interruption, but I think the slides are not moving.
They're not moving? They're moving on my side.
It's not in our...
Oh, sorry. Is it moving now?
No.
Let me stop sharing and share again.
Yes, please.
Okay, sorry about that, but it's moving on my end.
It's fine, it's fine. Yes.
Is it moving now?
Now we are on overview of diabetes mellitus.
Yes, and it's type 2 highlighted, right?
Yes, thank you very much.
Thank you. So our focus, as I said, is diabetes type 2 mellitus, which compromises majority of cases of diabetes, around 80%. So if we look at just a brief overview, what's the difference between the two? Type 1 diabetes, as you can see to my left, it's due to autoimmune attack of beta cells in the pancreas. So you lose beta cells, and this causes a complete insulin dysfunction. So you have no insulin produced. So this is absolute insulin deficiency. And this is considered juvenile diabetes with birth due to, as I said, some genetic factors. But on the other hand, when you look at diabetes type 2 compromises for about 80% of cases, which are in adults. Diabetes type 2 is different. It's due to some genetic factors and mainly lifestyle, which develops later on. This type of diabetes is due to many other factors associated also, but this builds insulin resistance. So there is secretion of insulin, but some resistance is built there, leading to hyperglycemia and different factors like increased lipids in the blood, inflammatory cytokines, and amyloid accumulation affects this resistance of insulin. So due to glucocytotoxicity and other factors. So today our focus will be on type 2 diabetes, not type 1. So every time we talk about diabetes, we talk about islets. So this is a section of the pancreas, as you can see here. Most people aren't aware what are islets. So islets of Langerhans are named under a scientist who discovered them. They compose about 1 to 2% of the pancreatic mass. So they are scattered around the pancreas. Roughly from the pancreas mass, you have 1 to 2% of this is islets of Langerhans. So what are these islets? As you can see here, they have some arsenal tissues. So you will find islets scattered in the tail of the pancreas and the head majority. So you can see here they have arsenal tissues, which is the outer layer. The inner layer of this islet is composed of different cells. Majority of the islet, as you can see here in yellow, is beta cells that secrete insulin, so about 65 to 85%. And then you have 20 to 25% alpha cells, which are glucagon producing cells, as you can see. Also, the rest comprises of different subtypes of cells, which is delta cells, somatostatin producing. You have the polypancreatic peptide cells, and you have gliarin cells. Well, in this endocrine compartment, they are considered the endocrine compartment, which is the endocrine cells of the islet. You have some capillaries, obviously, which supply these cells with blood to do their proper function in the pancreas. So when we want to talk about specifically diabetes type 2 mellitus, many studies looked at cellular dysfunctions and what is going on with diabetes type 2 mellitus. They hypothesized that the cellular dysfunction in type 2 play an important role in the disease pathogenesis. So when I say cellular dysfunction, basically this can be due to glucocytotoxicity, can be to loss of mass of these beta cells, can be due to beta cells apoptosis, and so on. Also, studies looked at amyloid accumulation and how it affects these beta cells. So also studies looked at metabolic stress, especially in type 2 diabetes. We all know that this is due to metabolism, stress, food diet, and lifestyle. So this metabolic stress can cause impaired insulin secretion. There are very important transcription factors, which has to do with these beta cells, maturity level and regulation of pancreas development and insulin gene secretion. The most common transcription factors that are studied are PDX1 and NKX2.2. Even in the family of NKX, you have NKX6.1. So basically, these regulators work, their transcription factors, they play a role in the fate of these beta cells. For example, PDX1 is a master regulator of the pancreas development and beta cell identity and insulin gene transcription. When we look at NKX2.2, this works in coherence with PDX1 to ensure that these beta cells are mature in their identity, and they repress other genes like alpha cells that produce glucagon because in a fate for them to produce insulin instead of glucagon. So basically, this is just one image that shows if you have a loss of PDX1 in any diabetic phenotype, so this PDX1 plays a role in beta cell identity and production of insulin, as you can see here, less glucagon and less other factors, transcription factors like MAFA-B and RX. What happens is when there is a loss of PDX1, which is a transcription factor, you can get sometimes negative effects. The cell can transform from being a beta-like cell to an alpha-like cell, producing more glucagon, more MAFA-B, and less ARX. So these transcription factors play an important role in the diabetic phenotype. So the cell does what it's supposed to do. For example, with beta cell, it does what it's supposed to do for beta cell identity to produce insulin. Any defect in these transcription factors can affect the cell fate or can cause the cell to differentiate into alpha cells. So our topic today is talking about a specific transcription factor, which is transducin-like enhancer split TLE. This is another family protein of a transcription factor which maintains beta cell identity in correlation with other transcription factors, like I said, PDX1 and KX2.2 and 6.1. So what does it do? Basically, it's a co-repressor. When I say a co-repressor, meaning it should repress alpha cells. Why? Because its role is to maintain beta cell identity to produce insulin. Well, in pancreatic beta cell, the most common TLE proteins are TLE1 and 3. As I said, they're crucial to maintain the cell identity, promoting functional maturation, and preventing de-differentiation. When I say de-differentiation, meaning a cell changing from a beta cell-like to an alpha cell-like. So there have been a lot of studies done in adult mice. Some of them are in vivo. They looked at the role of transcription factor TLE3 and 1 in maintaining beta cell identity. However, when we look at human studies, the role of TLE3 is not detectable. Most of the studies have been done in human on TLE1, which is the transcription factor that we're going to look at today. So what is TLE1? TLE1 is a gene. Actually, this gene is very close to type 2 diabetes lupus, which is referenced in its location and the number you can see here. So this gene lies very close to the locus of type 2 diabetes mellitus. So what we tried to do in our study, we tried to explore the expression in human islets. So we are looking at human islets and what is the expression of TLE1 between two different type of donors. Here, our control was non-diabetic donors versus type 2 diabetes mellitus donors. So what was our approach in this study? If we look at the methods, I'm not going to go in detail into the methods. I'm just going to give you a brief overview of the methods. So basically, we had pancreas donated from an isolation center in Canada, where they have a isolation lab. So they received the tail of the pancreas. The tail of the pancreas was fixed in blocks and sent to us so we can do our different islet studying for two different subcategories. So these donors were 7 donors non-diabetic and 7 donors that had diabetes type 2 mellitus. The different methods we looked at to study the insulin and the alpha cells and TLE expression was immunofluorescence. We also looked at the quality of imaging and how to analyze them after doing the staining. We also looked at a validation tool, which is a cell culture using cell line and transfection of these cells to study knockdown of specific TLE1 gene. We also did some RNA extraction, cDNA, and RT-PCR to study the level of expression of TLE and glucagon. So if we look at the first table with the results, you can see here you have 7 donors for type 2 diabetes. They are mentioned by donor ID, okay? And you can see in the control you have 7 donors. Each donor should be given an ID. These donors were matched for age, okay? Because of course, if you want to match donors, they have to be matched for age and BMI index, because any variation in BMI index and age can affect the results. The HbA1c is present here, you can see in percentage. So when we looked at mean number of islets to assess in the diabetic donors, we looked at 42 islets versus 48 islets in the control. The mean age for these donors were indicated, and they were all matched, as I said, for age and BMI index. So this was the criteria for the donors table, receiving the tail from the pancreas. Once we received the tail from the pancreas, we isolated these islets. These islets isolation, I'm not going to go through the process. It's a long process to get the islets isolated, and then embed them in blocks for us to do our specific staining. So as you can see here, this is an immunofluorescent staining for these islets in the sections of the pancreas. So panel A on the top, as you can see here, is non-diabetic control, and down it's type 2 diabetes mellitus, panel B. So let's look at this. As you can see here, I have the circular structures are my islets, okay? And this is the niche of the pancreas, which is stained in blue, okay? Which resembles the nucleus. We call it DAPI stain. The green that appears in the staining is insulin, and the red appears as glucagon. As we look at the non-diabetic panel, you can see here that insulin expression is very clear. Okay, these patients do not have diabetes. And the glucagon expression is faded, so it's less glucagon present. When we looked at the diabetic panel, which was type 2, you can see an increase here, as you can see in glucagon expression in compared to the normal panel. Well, when we look at some specific islets, so if you see bottom in panel B, the first picture, there is a box there. So we highlighted some islets because we saw a bihormonal expression. What do I mean when I say bihormonal? Meaning, if you look at these images here, this is a cross-sectional image here, you can see some cells in the type 2 diabetes express insulin and glucagon at the same time. So the red again is glucagon, the green is insulin. You can see it clearly here, this box, that some cells express both at the same time. And if you went through the paper, we separated the images to see the insulin expression versus the glucagon expression. And these are cytoplasmic expression, they are not nucleus expression, as you can see here, because the nucleus should be stained with DAPI, which is blue. So these are images taken by a specific microscope. They are 3D images. So we notice a bihormonal co-expression in type 2 diabetes for insulin and glucagon. So what was our next step? Now we get donors, okay? We have to ensure that my normal donors and the type 2 diabetes donors have equal number of cell distribution. So the graph to your left here, I know it looks complicated, graph D. These are number of each donors. As you can see, the red is for type 2 diabetes, the blue is for normal. We made sure that the sum of the endocrine cell, when I say distribution number of endocrine cell, which is sum of insulin producing cell, plus glucagon plus bihormonal, are comparable across all donors. As you can see, they are roughly in the average of 50. Some cells are just outliers, but majority are comparable across all islet cells in all donors. Each of these dots represent a cell in the islet, but they are comparable in terms of number, in terms of the average. So the sum of these endocrine cells. When we looked at the average, if you look at the graph to your right, which is E, when you look at control versus type 2 diabetes, each circle represents an islet. So for the control, you have 7 donors. You can see the dots. For the black one, you have also 7 donors. You can see that the average endocrine cells, when I say endocrine cells, I mean glucagon, insulin, which is our interest, and the bihormonal, they are equally the same. So there is no difference in the number of cells of these islets in the non-diabetic versus diabetic. Roughly in the control, they are around 80. And in type 2 diabetes, roughly in the control, they are around 83. and in type 2, they're around 80. So we have the consistency across in the number of endocrine cells for our study. So what was our next step? So now we looked at insulin, we looked at glucagon, we looked at the consistency of the number of cells. We went ahead and checked our TLE1, which is our transcription factor in relation to beta cells. We wanted to see what is the role of TLE1 in relation to insulin producing cells. So if you can see here, this is a non-diabetic donor, okay? We highlighted one of the islets here. And we stained, you can see again, insulin is green, glucagon is red, TLE is white, okay? When we stained for TLE3, or TLE1, sorry, because these are human islets, not mouse. So you can see here that TLE1 is mainly present in insulin-producing cells. You can see the green in conjunction with the white. So TLE1 is highly expressed by beta cells, insulin-producing cells. There are studies that said TLE1 can be expressed by alpha-producing cells, but it's very low. So it predominantly expresses beta cells that produces insulin. When you look at the glucagon panel, these TLE1, which are white, they don't express much glucagon, very less. We also noticed in this image that TLE1 is localized in the nucleus of these islets and in the exocrine compartment, exocrine meaning the outer compartment of the islet, okay, and in the nucleus. So when we did our calculation for percent of TLE in terms of insulin-producing cells, majority of TLE is expressed by beta cells, which are insulin-producing cells. Very few TLE cells, which is below 1. You can see the average here. The line here is below 1, produces glucagon. If we talk about bihormonal, insulin plus glucagon, very less amount of TLE1 produces bihormonal, which is about 0.7. Now, this is an image, keep in mind, of a non-diabetic donor, okay? So this is a non-diabetic donor, which is a normal person. So in a normal scenario, this is how it should be. Now, when we compared our different type of islets in the control and the type 2 diabetes, for percent of insulin, how much insulin is produced for both of them, we noticed that when we looked at control versus diabetes, the percent of insulin per islet cell, you can see here in the control is higher, okay? In the diabetes is a bit less, so we got a significant difference. This line indicates a p-value of less than 0.05, that meaning there is a significant difference between percent of insulin per islet in type 2 diabetes and control. Then, when we looked at glucagon percentage, which is another hormone in the islet, we looked at glucagon percentage. We realized in the type 2 diabetes that the glucagon percentage is higher than the control. So here you can see there is a significant difference alone for glucagon between control and type 2. Then if you move on, we said, okay, let's look at cells that co-express. When I say bihormonal, meaning they express insulin and glucagon at the same time, we notice a huge significant difference between control patients and type 2 diabetes. As you can see, the control here, I look at this mean line, it's below 1. Here, it's above 2. So there are some cells that become bihormonal, but are they really expressing both at the same time, or are they de-differentiating? Meaning, when I say de-differentiating, meaning are these beta cells changing into alpha cells, or what is happening here? So what we decided to do here, we decided again to do an immunofluorescence staining. The top panel A is my control, okay, as you can see. The bottom panel here is an islet, which is a type 2 diabetes islet. You can see clearly here in this panel that the TLE expression in a normal, which is in white, is more apparent than TLE1 expression in a diabetic. If we look at the glucagon panel, again, this is confirming our findings previously, that you have more glucagon expression when you lose TLE1. In a normal scenario, when you have TLE1, it doesn't express much glucagon. But when we look at the insulin expression here, we didn't find much of a significant difference in the insulin expression. So if you look at the graphs in the bottom, percent TLE expression per islet for control, of course, is higher than type 2 diabetes, which I showed you here in the image. If you look at percent insulin expression for TLE1, insulin expression with TLE1, you can see here there's no much of a significant difference between type 2 diabetes and control. Here the line comes at almost 60, 50 something, and here the line comes at 42, I think, or 43. Now, we decided to do a regression model to see what's the ratio of alpha to beta cells in terms of each islet. Each of these dots are my islets. The white one, which is not highlighted, is control. The black one is diabetes type 2, as you can see here. We did a regression plot. As you can see in this regression plot, as the TLE expression decreases, the number of cells that are changing from the ratio of alpha to beta cells increases. So I'm getting, as my expression of TLE1 goes down, I'm getting more alpha cell ratio in compared to beta cells, okay, in type 2 diabetes. And you can see from this regression plot with the R value of minus 0.56. The total number of islets that were assessed here to do this regression model was 633 islets to ensure that you have an increase in the ratio of alpha cells in compared to beta cells in type 2 diabetes, as you can see in this regression. So this is an inverse relationship, meaning as I decrease my TLE expression, I'm getting the ratio of alpha cells are increasing in compared to beta cells, which explains why here I'm getting more glucagon secreted. So the expression of glucagon here is increasing. Now, we try to see what is TLE1 really associated with glucagon co-repression. So in a normal case, when I have TLE1, glucagon should be co-repressed because this is a transcription regulator, which we showed before that it's mainly associated with beta cell, meaning with insulin secretion. What we did is we had a human cell line, which is a purchased beta cell line, which is abbreviated as human Indoc BH1 cell line. We said, okay, let's see the effect, let's study the gene expression. When I say gene expression, meaning in a cell line, not in the human islets. Now I'm moving to a cell line to show a functional study to show, is it really TLE1 related to glucagon gene expression? So what we did is we purchased a cell line. We bought two silencing RNA. We used two different methods, as you can see here on the top. I'm not going to go into the nitty-gritty detail. I'll just go general so everybody gets a gist of it. So if you see these graphs on the top, we had two silencing RNA methods that we used for these cells to knock down TLE gene. So when I say knock down, you can see here, this is my scrambled control in black. Okay, and these were normalized for a reference gene in the PCR. And you can see I have two different siRNA methods to knock down this gene. We tried two different purchased methods of transfecting the cell with silencing RNA to knock down the gene. We did a PCR to confirm that this gene expression is decreased. So this panel A to my left shows me my control. and the number of fold increase here in the control is 1. In the probe 1 and 2, which is a 2 different siRNA methods that we use to confirm that we knock down TLE1, we can see here very clearly that the TLE1 expression is decreased. But probe 2, which is a different method used, decreased the expression more. So we chose probe 2 because it worked more efficiently to silence the gene and knock it down. So now this picture to your right in B again is my PCR normalized to a reference gene, which is your RPLP0. You can see here, once TLE1 was knocked down in the control, because TLE1 was not knocked down, the glucagon expression is less. Once I knock down TLE1, there is a significant difference in glucagon expression as it's increasing. You can see it very clearly. This is exactly what was happening with the islets here in terms of increase of glucagon expression due to decrease of TLE1. So mRNA expression of glucagon, because I'm talking about PCR here, is increased. When we looked at the images, I'm sorry, maybe some of these images are not very clear. The first panel on the top here is my control, okay, without any knockdown. You can see here insulin is clear. There's not much glucagon in the green panel, and there is a lot of TLE1 because this is a control without any knockdown. The siRNA TLE1 knockdown, which we chose probe 2, you can see clearly the TLE1 expression is decreased in the staining here. It is much less here. You can see we circled some cells. Insulin expression went down slightly, although it's not apparent here, but it went down when we calculated it per protein. So you can see clearly the cells circled here in the circle. Some of these cells that are expressing insulin, some of them are expressing also glucagon. Circled, you can see the green, they're expressing glucagon. Some of the cells also, the ratio of alpha cells, as you can see here in the bottom in E, you can see a bihormonal expression, but also you can see the ratio of glucagon here increasing in the green circles in the bottom. These green here, as you can see in the last panel in the bottom, which is E, the green here of glucagon is apparent with some cells that are producing insulin. So the ratio of glucagon here is apparent without any TLE. You can see here these circles have no white dots whatsoever when we zoom in those areas. So this was just a functional study to prove in a cell line, not in human islets, I'm repeating again, in a cell line, that TLE inhibition or knockdown can cause an increase of expression of messenger RNA glucagon, okay? And this knockdown sometimes increases the alpha cell to beta cell ratio because due to the absence of TLE1. So we proved this in a cell line model. So our concluding remarks is nuclear TLE1 expression is predominantly with insulin-only positive cell of islets because as we saw in our study that majority of TLE1 is localized with beta cells, around 98% actually. And the problem is with patients with type 2 diabetes, this TLE1 expression is reduced. So when this TLE1 expression is reduced, we get a higher alpha to beta cell ratio. And because TLE1 is localized with insulin producing cell, we consider it to be monohormonal in human islets. Okay, so when you increase this ratio of alpha cells in compared to beta, you get an increased abundance of bihormonal cells in type 2 diabetes mellitus patients. And we confirmed this by doing a functional study on a cell line that TLE1 should be a glucagon repressor. And this was supported by our knockdown studies with the human cell line. and we saw that reduced TLE expression leads to glucagon depression, okay, increase in glucagon. So our conclusion demonstrates that these findings demonstrate that TLE expression is distributed in type 2 diabetes mellitus in association with increased glucagon positive cells. So there is an increased glucagon positive cells, potentially reflecting a change of fate, meaning these beta cells are converting to alpha cells, potentially a de-differentiation in the fate of these beta cells. So when you get a lot of alpha cells, you will get a lot of increase in blood glucose. And this is what type 2 diabetes patient might be experiencing. This is just a window of a molecular aspect in relation to a TLE1 transcription factor. And this is not the only factor. We all know that there are many other factors involved in this disease to cause serious complication. Thank you very much. I would just like to acknowledge the team that worked with me, which was part of the university and Ministry of Health. And thank you very much for your attention. I always like to put an islet picture because most of my work was a lot of immunofluorescence staining with islet cells. This is actually an actual picture from one of the stains that we've done in one of the human islets with different nuclear stains. The green always resembles insulin, and we looked at different compartments. Thank you very much for your attention and listening. Thank you very much.
Thank you very much, Dr. Najwa, for a very fruitful presentation. It sounds like a very, let's say, complicated study, but it seems that you have done it very smoothly and presented it also in a very clear way. I have one question before going to the audience. As a researcher, do you see that TLE1 as a potential therapeutic target, and what challenges would happen to be addressed before translating this wonderful findings into the clinical practice?
Thank you very much for your question. It's a very valid question. When I will go back to trying, maybe people were wondering why we didn't knock down this gene in the human islets.
Yeah.
When we go back to the factor of human islets, it's very tedious to deal with because even when we try to transfect the cells and knock them down, it was a process of a lot of optimization that requires more other aspects into it. So I would say for clinical, it's not just easy to focus just on TLE1 and translate it into clinical. There are many other factors that we need to look at overall for as therapeutic target for type 2 diabetes, because down the road, when you look at the whole signal transduction and the maturity of these cells, there are many other transcription factors that should also be studied in correlation to TLE1, which I spoke about, which is PDX1. Downstream, you have NKX 6.1.
Yeah.
So you need to study the whole aspect of how can this affect the fate of the cell. Second of all these studies were done in vitro, meaning outside. So these are human islets got from donors, and they are done outside. These studies would be done nicely if they're depicted in an in vivo model, inside a mouse model, right, and see what are the fates of these cells in a mouse model if this TLE1 is knocked down. How can it affect other factors? Because when we talk about in vitro models, it's easier to mimic what is not the actual condition in an actual human body. Thank you very much.
Right, clear enough. Thank you very much, Dr. Najwa. And I would like also to invite Dr. Shadia Al-Bahlani to add from her expertise to tell us on the challenges and the reality of translating such a research finding into a real clinical practice. So, Dr. Shadia.
Assalamu alaikum wa rahmatullahi wa barakatuh jami'an. First of all, I would like to thank you and thank the organizing team for inviting me for such a wonderful talk. It's a very excellent piece of work. I really convey Dr. Najwa and her team. And I worked on confocal microscope, on histology, paraffin blocks, RT-PCR, and I can tell you that it's not an easy experiment to conduct, and there is a lot of challenges. Regarding your question, Dr. Najwa answered it perfectly. We always start with the in vitro, where we have our idea or theory and try to test it in cell lines. And then we upgrade it to the in vivo in animal studies. And then we go later on for patient samples and clinical trials. So it's we can consider it. I always teach my students that research is a big puzzle, and each researcher will add a piece until the whole picture will be completed. So definitely this is make a good base for further experiment.
Thank you very much, Dr. Shadia. Mr. Nibras, you have a question, as I'm seeing in the chat box. Maybe you can unmute yourself and participate in the discussion with your question, please.
Sorry, I had some questions, or I am not allowed to ask.
No, you are welcome. Please.
Ask, Doctor. Go ahead.
Yes, please.
Okay, so most of these questions is, just to enhance the research, and I know that you published it in 2020. I'm sure that there is more have been published, and maybe the team continue to work on the same aspect. I was just wondering that I know it's really hard to get patient samples or donors, but when you mention that non-diabetic and you were referring it to normal or control, are you sure that there is no any comorbidity with those patients?
Thank you very much. That's a very good question, Doctora, because see, these donors, we received the donors from an islet center from Edmonton. This was not very clearly addressed in terms of comorbidities. This is a very valid question in terms of what other complication that these type of patients have. And if you noticed in one of the donors table, the HbA1c was missing for one diabetic donor. So in the register, they take as much as they have an islet core center in Canada and in the UK. They take as much as they can to ensure that there's no other comorbidities, but you don't know, it depends again on the register how these patient comorbidities were taken. But and the other challenge is to get these donors. As you can see, the number is not very high. We're talking about 7, but in terms of number of islets, it's good for the study. But to get such donors also sometimes also is a challenge. But majority of the non-diabetic most of them don't have complication, but I'm sure there are some other minor complications that are not addressed in the register.
Okay. I also noticed that the mean age for both groups are around 7 years, except one for 15 years.
Yes.
And it's considered as
about the control or the diabetic?
Yeah, the diabetic. So I'm not sure, because those considered as short disease duration. Do you think it might have some influence on the quality of those, islands?
Yes, definitely, because some durations were not long enough, which is 0.1, 0.5, and the other one was 2. This is a very valid question, and which was, we addressed it, which is R064 and R131. They were not a high duration. And when you compare something that is not a high duration, it can affect, the quality of the study, because maybe even in terms of when you saw that we saw no significant difference in the insulin expressing cells between the normal and the type 2, some not long duration of diabetes can affect the results, can a bit skew them. But unfortunately, this is what we received in terms of duration, and that's a very valid point. Thank you.
And do you think the gender differences might influence? Because you are comparing sometimes male to female. You couldn't get a lot of female in your second group.
Yes, definitely. We tried our best just to match the age and the BMI index. Mainly BMI influences a lot of function, even in terms of beta cell and insulin secretion. But of course, the gender makeup also makes a difference, female body versus male, and the characteristics. But this is what we got from the donor center. Most male patients we got rather than female.
Regarding the knockdown, it's an excellent experiment, and it's
very
considered like
one.
I know it's really hard to knock down genes,
Yeah.
But I was wondering why you didn't check or identify downstreams like BCL2, or since it's a transcription factor, it will have a downstream, just to make sure that the effect is also functional, because you didn't detect the protein as a content. You just detected as immunofluorescence, which is considered just to show localization, but it's not the exact one that will give you the content of the protein.
Yes, that's a very valid point. Normally, we do functional studies for glucose secretion in relation to the protein content. So we can see how the expression changes with the ELISA versus Bradford in terms of protein content. But we had a lot of imaging and analysis to do. That's a very valid point. And make sure the analyses are blinded, and then 2 different independent analyses. And sometimes the quality of the islets that were received in some sections, they were not so great, because we're talking about human here. But that's a very valid point. We didn't get time, unfortunately, to look at downstream genes and validate.
You can apply it in different cell lines.
Yes.
And you can also do the opposite. You overexpress the gene. And then
there are some studies, Doctora, that did overexpression in mouse models for TLE1. They were done in mouse models for overexpression. But also it would be useful. We were struggling to knock down, to be honest with you, TLE in the human islets. It was like a mission impossible. At the end, all the optimization didn't work.
Okay. Well, alhamdulillah, what you did so far is fair enough. My last question, to give the floor to the audience, I don't want to be very selfish. In the immunofluorescence, you always keep repeating localized and co-localized. Did you try to check the interaction between the proteins?
No, unfortunately, no.
Because I could see in one of the images, there is like a yellowish stain.
Yes.
Which indicate, that the red and the green might give that yellow. But still, we need more experiment to confirm it. Thank you so much.
Thank you, Doctora, for your valid.
Thank you for the presentation and for the, the explanation. You really explained the study in a very nice way. Thank you so much.
Thank you very much, Dr. Najwa and Dr. Shadia. That was a great discussion. Now we'll give a chance for the audience. So, Mr. Nibras, would you please unmute yourself and ask your question?
Assalamu alaikum.
Alaikum salam.
I believe most of my questions has been asked by Dr. Shadia here. But if I would like to highlight something, thank you, Dr. Nagwa, for the informative informations. But my main question could be about the genotype of the locus itself, since we know this is a locus and it's mainly related to the functionality of Iceland land. Have you investigated the genotype of those locus, especially in the donor cohort?
No.
Consider the small number of the samples and how the genotype will affect?
That's a very good point. No, we didn't investigate that aspect, because our concern was trying to prove the transcription factor TLE1 role in these 2 different type of donors. But no, we didn't look at it. This would be a very good addition to look at the different genotypes. No, unfortunately.
Okay, so that's really good angle to investigate then, especially if you have the data, so we can work on that.
Sure. the genetics speaking here.
The second question, Doctor, I couldn't get why the bihormonal cells did not show the same reductions in monohormonal beta cells exactly.
Yes, okay. What happened? So your question is why the bihormonal are not showing? Because we thought there is something like a dysfunction in the TLE1 because, you know, TLE1 should express, it is monohormonal. It's mainly predominant, as I showed in beta cell, to express the, it's mainly predominant to express insulin. I think, see, the bihormonal, I believe so. There is a bihormonal which doesn't have TLE. They didn't have TLE expression. They were a lot of cells that are bihormonal. But the reason why I believe that we didn't have probably when you look at the donors itself, the duration is not the same. Some donors had less duration of diabetes. Some donors had higher duration. And then the type of islets in the sections also, when you scan the tail itself, you have different islets with different morphologies. Some quality after embedding are ruined for the islets. So I don't think it's, I totally believe, I don't think also it's a bihormonal, more of a de-differentiation, meaning you are getting a raise in alpha cells rather than a raise in beta cells. So meaning the beta cell is changing fate. So because TLE role is to co-repress glucagon, but here you have a change of fate. The debate always is like, I think we didn't have enough bihormonal expression. Again, this refers to the number of islets and how many are present and how many were preserved after fixing also, and the sections. But you can see some of them were like bihormonal with no TLE1, but it's more of a, I feel, increase in alpha cell ratio than beta cell ratio.
So is this something that we can modify it if we rerun the experiment by increasing the sample size, for example, or it's something biologically we cannot fix it? Or
see, this bihormonal aspect is debatable here because some people say there is a bihormonal expression. Some people say no, it's more of a change of fate of alpha cell ratio to beta cell ratio. And most studies say that TLE1 is a change of fate from alpha cell, from beta cell to alpha cell. You get an increase in the ratio. There are some cells that will be bihormonal, but the number, I didn't see a study yet that showed there's a lot of bihormonal. I saw most of studies talking about a change of fate, mainly, to be honest.
Really nice. Thank you, Dr. Nago. Thank you.
Thank you.
Thank you very much, Mr. Nabras. And now we'll give a chance to Ms. Taimura. Would you please ask your question? Ms. Taimura.
أيوة السلام عليكم.
Taimura, sorry.
السلام عليكم. Thank you, Dr. Najwa, for your informative presentation. Just I have one question. The type 2 diabetic type 2 treatment, we know that it alters the islet cells
Yes,
structures.
you are talking about metformin?
Metformin and the GLP-1 receptors.
GLP-1, yes.
Insulin,
Yeah.
Okay.
Yeah.
So we know they alter the structures of the islet cells and also the cell stress level. So
Yeah.
did you account this for the medication history, duration of the disease, and even the terminal glycemic status of the type 2? Could that lead to the reduction of the TL1 expression, or the TLN reduction in TLN could be as a pathogenesis? Yani?
Most studies showed that the actual reduction, even there was a study done with actual patient samples for the fate of beta to alpha cells, which was, if you look at reference 28, which was done with European subjects with type 2. They see there is an increase in the mass of the alpha cells regardless in terms of medication toxicity affecting or not. We all know that medication has its own effects on different parameters.
Yes.
And different aspects of any type of disease. But no, most studies prove this is more of a molecular aspect rather than, even some studies were done on the human samples, which, like I said, European subjects, they noticed that the mass of the alpha cells are higher regardless, meaning there is something that is causing this mass of alpha cells to be high. We also related in this study other transcription factors. I didn't show you the whole picture. We related other transcription factors downstream, like PDX1, NKX6.1. They are also affected downstream in type 2 diabetes. These transcription factors are also involved in beta cell maturation, beta cell identity. Each transcription factor has its own role. So NKX6.1 is downstream to TLE1. So with type 2 diabetes, you will notice even when we took the human islets, we stained them, there's a lot of transcription factors that are expression are decreased. So I think it's more of a molecular aspect rather than a drug toxicity, to be honest with you.
It's more of pathogenesis.
Yes. Yeah.
Yes, thank you. And also for the bi-hormonal, the transition phenotypes, the study is reporting like a doubling of the insulin glucagon bi-hormonal cells in type 2. And however, the vast majority of these bi-hormonal cells is found to be TLE negative.
Yes.
So if TLE downregulation drive this transition, so why TLE already is absent, like in small fraction of bi-hormonal cells, which is found in the healthy whole control?
I understand your point. We thought the bi-hormone, we were also a bit shocked about this, but we thought it's not really, as I was explaining to Mr. Nibras, it's not really bi-hormonal. It's a change of fate. It's more a de-differentiation, meaning you are losing, and especially if we had a study that, as Dr. Shadia illustrated, compare it with protein, it would have been better maybe to show, to normalize the protein expression so we can decide explicitly. But I think it's more of a change of fate, increasing the ratio of alpha cell
to
beta rather than it be bi-hormonal. I think the bi-hormonal needs to be investigated further given the conditions we had and the limitations we had in terms of donors, to be honest with you. And you can see also the duration versus other aspects. Thank
so
you
it could be like a trans differentiation between
yes, it could be, yes. Because in literature, when they talk about TLE1, they talk about it being monohormonal, mainly. Yeah, most literature, and you can see from what we showed like percent of glucagon is very low in normal and percent of bi-hormonal. But then we didn't get that feel of a lot of cells being bi-hormonal. There is a feel, but there was some difference there, but I think we still need to investigate further, actually. Yes.
Thank you very much, Dr.
Thank you.
Thank you very much, Dr. Taimoura and Dr. Najwa. Dr. Najwa, we will move now to the chat box. We have a question from the Dean Office. And the question is stating like, how do you address the issue of islet size, heterogeneity, and variation in cellular composition, especially in type 2 diabetes slides?
Okay. This is a very good question, and it's always challenging when you actually...
Dr. Najwa, we can't hear. Yeah.
Yeah, thank you for the question. This is a very good question, and it's always challenging when we look at human islets in terms of different sizes, different heterogeneity, different cellular compositions. So what we tried our best to do is when I showed that graph, which showed that the compartment of the endocrine cells, the sum of insulin cells and glucagon and bi-hormonal, they are not changed across the donors. They are consistent. Even when we showed the graph that showed the control versus the diabetic, the average endocrine cells, there was no significant difference. Heterogeneity is bound to happen there because we are talking about islet cells, human islets. But also when these cells go through isolation protocol, the isolation protocol also it's very strict. They study the purity of these islets. They study also how there is a different team that deals with the isolation protocol and how pure are these islets, how viable are these islets, and different scoring aspects of these islets to meet as much as we can the study outcome, which is the islet core lab in Canada versus the core lab in the UK. It's a whole facility, actually, because also you have to remember these islets also get transplanted into patients. So also you have to go through the proper protocol to ensure all these aspects. But thank you very much. We can do as much as we can, but at the end, these are heterogeneous cells. Thank you for the question.
Thank you very much, Dr. Najwa. Dr. Saeed?
Yes, I do. But there's someone else, Zainab. I think Zainab Talib, she asked
Yeah,
question.
she is the last one in the chat box.
Okay.
Zainab Talib is asking, considering the small sample size, can we conclude that reduced TLE1 expression occurs in all patients with type 2 diabetes, or it is limited to a specific group of patients? This is the first question. The second question is the increase in alpha cell number associated with reduced TLE1 expression due to beta cell transdifferentiation or simply a loss of beta cell identity without a complete cell fate switch?
Okay, so I will go with the first question.
Yes, please.
Can we conclude that reduced expression? I would say majority, because I will not say all, because I don't know. This all depends on different types of patients' conditions and comorbidities and what other complications these patients have. But majority of studies have shown that there is a reduced TLE expression in type 2 diabetes patient. And it's very difficult to say all patients here because it depends on the comorbidities and what complication these patients also have. So when we talk, is the increased alpha cell number associated with reduced TLE expression due to... Now, we say the most aspect is change of fate of these beta cells, meaning you're getting more alpha cells expression, which we call it mainly de-differentiation. We did some de-differentiation studies to ratio increase in terms of beta cells. And some aspect of can be change trans-differentiation, but it's more of de-differentiation. See, when you talk about loss of beta cell identity, these are all interrelated, by the way. If TLE1 is responsible for beta cell maturation, stopping beta cell from de-differentiation, it's a co-repressor for glucagon, meaning it maintains the beta cell identity, because that's its role. If you cannot maintain this beta cell identity, which is the role of TLE1, these other things will happen. So these are two interrelated things. They're not looked at separately. Beta cell identity in relation to a co-regulator or co-expressor, which is a transcription factor.
Thank you very much, Dr. Najwa. I think we have addressed all of the questions.
Sorry.
Yes, Dr. Said.
Okay. Firstly, I would like to appreciate all of you, particularly Dr. Fatma. Dr. Fatma was awarded the best presentation during international conference. I think it is in higher education that was conducted 2025. Now, also appreciate Dr. Najwa, mashallah, and Dr. Shadia. I'm not familiar with the terms used in lab, but one word attracted me a little bit, and I would like from Dr. Najwa to elaborate. What do you mean by knockdown? It was explained with the behavioral perspective has a different meaning, but within the lab, it has a different meaning. This is number one. Number two, what is your future plan? Are you plan to collaborate with other specialty, like, for example, behavior scientists, to collaborate with them and conduct a multidisciplinary research?
Okay, when I say knockdown, or I use the word knockdown because we use it scientifically, meaning I use a technique, which was the silencing RNA, to stop the function of this gene. Meaning, I stop the function of this gene, I knock down this gene, meaning now this gene is not functioning. Okay. So I want to see when the gene is not functioning, what is happening at the messenger RNA level to the glucagon expression in related or insulin expression in related. So at a gene level, what is happening when I stop the function of a specific gene? Clear?
Yes, clear. What about your collaboration with the?
See, I think we are, Oman, in terms of to run these studies, also even for my PhD studies, we need donors, and we need a proper center to do all these isolation and whatnot. I think we are really here at a very early phase, because the program for organ donation just started recently, and most of organ donation now it's done for clinical purposes. We didn't see anything yet for in terms of research purposes. This was my actually future plan. I was waiting for the organ donation program to start, but I think such studies can be implemented. But again, given that we have donors, we can work on such studies in terms of cell line, to look at different cell lines, which we can do some collaboration studies with SKU and whatnot for cell lines. But in terms of donors and actual islets, this is a very complicated process, by the way. It needs a whole GMP facility. When I talk about GMP, this is a whole facility with a team who is trained properly to isolate these islets from the pancreas and ensure their quality is proper, their viability, their functioning properly. Because, once also you put them into the body, if we are transplanting into whatever it is, if the quality is not the standard scoring, as they asked about the heterogeneity and all that, if it's not up to standard, none of this will work. So I think we're still far ahead. We are still far behind. We still need to develop such protocols, but I think we can work on human cell lines.
Thank you so much, Dr. Fatima.
Thank you very much, Dr. Saeed, for your question, and thank you very much, Dr. Najwa, for answering a huge number of questions. But it points that actually you have catch up the interest of the audience, and your presentation was very clear. And I would also thank Dr. Shadia Al-Bahlani for being engaged in such a great discussion, and all of the audience to be engaged in a very fruitful discussion. I know that tomorrow is a holiday and everyone is waiting for me to close. So thank you very much, everyone. And that's it. Thank you.
Thank you very much.
Thank you so much. My pleasure. وإجازة سعيدة إن شاء الله.
إجازة سعيدة للجميع.
شكرا جزيلا، يعطيكم العافية.
الله يعافيك.